Meiosis is a type of cell division where a diploid cell divides to form 4 haploid daughter cells. The purpose of meiosis is to produce gametes. During the G1 phase, the cell grows larger, makes copies of organelles, and makes molecular building blocks for later steps. Depending on the type of cell it is, it may exit the cell cycle to a G0 phase or move on to DNA synthesis. During the S phase, the cell synthesizes a complete copy of its DNA and the centrosome duplicates. During the G2 phase, the cell continues growing, makes proteins and organelles, preparing for cell division. During prophase 1, the chromosomes condense and homologous chromosomes pair up. During prophase, crossing over may occur. Crossing over is where homologs exchange part of their DNA. After the synaptomal complex disintegrates, the chromosomes are connected by chiasmata, which are the sites of crossovers in chromosomes. This leads to the formation of chromosomes with unique allele combinations. During metaphase 1, homologous pairs line up at the metaphase plate and the two homologs bind to microtubules from opposite poles. The random orientation of homologous pairs leads to the independent assortment of chromosomes. For example, the homologs could have lined up like this, and these would both lead to different gametes. Both of those orientations are equally possible. During anaphase 1, the homologous pairs are pulled apart and moved to opposite ends of the cell. During telophase 1, chromosomes have moved to opposite ends of the cell when cytokinesis happens. The final result is 2 haploid daughter cells with duplicated chromosomes. Meiosis 1 is now complete and cells move on to meiosis 2. No DNA replication occurs before the second set of cell divisions. During meiosis 2, the sister chromatids separate. This process is very similar to mitosis. In prophase 2, a spindle apparatus forms and the spindle microtubules start to attach to the chromosomes. In metaphase 2, the chromosomes individually line up at the metaphase plate. In anaphase 2, the sister chromatids separate and are pulled to opposite sides of the cell. And in telophase 2, the chromosomes decondense, nuclear membranes reform, and cytokinesis occurs. The final product of meiosis is 4 haploid daughter cells. Meiosis creates genetically unique offspring because of 3 factors: crossing over, independent assortment, and random fertilization. Independent assortment produces 2 to the power of n possible chromosome combinations, with n being the number of single unique chromosomes, which would be around 8.4 million for humans. This results in around 70 trillion diploid combinations when these gametes fuse. This all accounts for the huge amount of variation in offspring. Thank you for watching.